Hello and welcome to Conversations on AFM. I'm Dr. Elisabeth Kugler, a science communicator and your host for today's conversation, the role of mechanical forces in health and disease. Today we're talking about the fascinating intersection of physics and biology to explore the fundamental forces that shape life. The research that we will talk about today uncovers how mechanical stress alters the material properties of living matter and influences the growth of cells and tissues. Our lovely guest today is Professor Dr. Elisabeth Fischer-Friedrich. She's the Heisenberg Professor at the Physics of Life Cluster of Excellence at the TU Dresden. She has a doctorate in physics and has worked at a multitude of prestigious institutions, including the Weizmann Institute of Science and the MPI for the Physics of Complex Systems. She's a winner of the Otto Hahn Medal in 2009 and the Hertha Sperner Prize in 2022. Welcome to you, Elisabeth.
Yeah, thanks for having me.
We're so excited to talk with you today about all things biology and biomechanics. So we kind of dive straight in, if that's okay, because your work is really highly interdisciplinary and it ranges from physics to biology and everything in between. And so the overarching aim is really to understand some fundamental questions of life. And we have quite a variety of different listeners. So their background might be biology, might be physics. But we thought to really set this scene, it would be great if you could introduce to them how mechanical stress influence the material properties of living matter and why it is important for biological function.
So one important way in which mechanical stress influences the mechanical properties of biological material is through stress stiffening or vice versa, stress softening. And stress stiffening relies on changes of the structure of the polymeric materials that most biological materials are made of. So we have stress stiffening, for instance, for extracellular matrix, collagen-based materials, for the actomyosin network. And this is also something that we know from everyday life, polymeric materials such as rubber. But mechanical properties are also in general important for biological materials, not only through their dependence on stress. So whether a biological material, cell or a tissue deforms when forces are acting on them depends on their material stiffness. So they might either comply if they're soft, or they might more or less keep their shape if an external force is acting, if they are stiffer. And cells and tissues are generating their own internal forces, often actomyosin based. So they could actually even tune their stiffness themselves through having higher and lower active stresses. High active stresses frequently giving rise to a stiffening, lower active stresses rather giving rise to a softening. And at the same time, they use these active stresses, of course, to deform themselves. So this is an interesting interplay. So you have more force generation, but you at the same time get stiffer. But then on the other hand, you also have timescale dependence in the material properties with a great tendency to become liquid-like on longer timescales due to molecular turnover. So we have here a very complex mechanical dynamical system that can give rise to quite a diversity in structure formation depending on the individual players, the time and then scales under consideration.
It's so amazing to hear that there's so many different aspects and things to consider. And I think for someone who hasn't done it, like myself, I think it's very difficult to understand how you even measure kind of stiffness and these biomechanics in cells. So could you maybe share with our listeners, how do you go about like measuring these biomechanical properties and how you maybe can use AFM or atomic force microscopy to help you with these experiments?
Yeah, so the concept is always the same, right? So we apply a deformation, typically a small one, and then we measure the force response of the cells, or like the forces necessary to do that, or vice versa. We apply small forces, and then through imaging, for instance, or just through seeing the height of the AFM cantilever, we read out the deformation of the material. And in order to interpret those experiments, one always needs an underlying mechanical model. So the most frequently used one for AFM indentation is the Hertz model, contact mechanics, and that assumes that we are simply indenting into an isotropic uniform material with a certain Young's modulus, a certain Poisson ratio, And so this essentially allows you to read out the mechanical stiffness necessary to poke into this material. So depending on what kind of indentar shape, pyramidal or spherical, you would have a different force response. But in a way, it's very simplistic. Usually people even assume that the material is incompressible. So we have a Poisson ratio of 0.5. This is convenient because then you can fit the force indentation curves of only one parameter. And that is often also more than enough. I mean, you also have the contact point that you're fitting. So it's not only one parameter, at least two. And this is also frequently more than enough to fit noisy biological data.
You're using a lot of assumptions from the physics field where you, for example, say cells are isotropic. You have assumptions that you kind of model the cell along a shell. You really try and make the cell itself as simplified as possible. And then you use measurement techniques to kind of not only understand how the measurement tool, like the cantilever, responds, but also how the cells kind of respond to that. And you probably use like comparative studies, like comparing condition A to condition B, and then understand how they both relate to each other using physics models.
Yeah, exactly. So, I mean, to break it down very much for lay people or people who are not into mechanics, at least. So we probe mechanics of the cells by poking into them, frequently using AFM indentation, or by squeezing them between two parallel plates. This could be also done by AFM, for instance. And from an intuitive point of view, you can already guess that this measures something like a material stiffness. Physics tells us, okay, there is not just one stiffness parameter. In general, there are more, at least two, namely one that tells us about the mechanical resistance to changes in the aspect ratio of a solid body, and the other one would be changes in the volume of a solid material. And then it can become arbitrarily more complicated if we have a composite system that is consisting of several different materials, like the cell is, right? It does not only consist of one material layer, but it's a composite material. We have timescale dependent mechanical properties, so viscoelasticity. We have nonlinear material properties, possibly. That means the stiffness will change depending on how much we deform it, how much stress is already present in the system due to active stresses, for instance. So it's a lot to consider. Sometimes it's easiest to say we just poke into it. We see how difficult it is to poke, what's the force necessary, and then we get at least a rough idea of how difficult it is to deform it. And that would give us the classical apparent Young's modulus that people most of the times measure when they do AFM indentation. And then you can get more fancy and say, okay, we at least recognize that, for instance, non-adherent cells, their mechanics is dominated by the actin cortex, so the biopolymer network underneath the plasma membrane at the cell periphery, or at least our studies have supported this idea. It's not the only structure in the cell that gives contributions to this mechanical response, but it's a very important one, at least if the deformations are small. And then the shell model is a good model with not that many parameters. So the one parameter would be the pre-existing tension, and then the second parameter could be the modulus that determines how difficult it is to increase the surface area of the shell.
Now we kind of want to switch to another topic, which is the basement membrane. This is a layer which lines the basal side of epithelial and endothelial cells, and it is important for the form and function of the surrounding tissues. We have spoken with one of our guests in episode 3, who was Professor Christina Kusser-Wierg, a little bit about the basement membrane and her research. But we would love to hear now from you how you study the basement membrane in your lab and what model and techniques you use to understand the basement membrane.
Yeah, thanks. So the basement membrane is a really exciting new topic that emerged in the lab, thanks to the collaboration with Professor Christian Dahn, who is a biology professor here also at the Technical University of Dresden. And he approached me and said, "Hey, Elisabeth, shall we not look into the basal membrane in the wing disc of Drosophila larvae?" And that's how it started. And by now we have done several studies, so not only in the wing disc of larvae, but also in egg chambers. But maybe let me start, or first also for the listeners. So basal membranes are specialized extracellular matrix layers that are attached to the basal phase of epithelia. So epithelia are sheet-like tissues in our body, so they're actually quite a prevalent tissue type. So leaving out blood cells, most human cells in our body are actually epithelial cells. And they are polarized, so they have a top and a bottom. And at the lateral faces, they are adhered to neighboring cells. So actually, so it can be multi-layered epithelia. I'm rather thinking about monolayered epithelia now, to be fair. And so the top, we have the top surface, which is the apical surface. Frequently, there is a prominent ectomyosin cortex supporting structurally the inside of the cell at the apical phase. And at the bottom, in layman terms, so the basal side, we have possibly also ectomyosin cortex, depends on the epithelial type, but we have at the outside the specialized layer of extracellular matrix. And it also contains collagen, more particular collagen 4, and then the other important structural protein there is laminin. And they are forming a complex network also with other associated proteins that are cross-linking, changing material properties, etc. So I found it interesting because it's like having an outer actic cortex. So initially we were super interested in the actic cortex. Now we learned about the structure, which is like an inverse actin cortex at the outside of the cell. And what is interesting is that opposed to the actin cortex, which actually turns over molecularly quite fast, so on the timescales of minutes, so to explain turnover, it means we have polymerization and depolymerization going on all the time. So the network never falls apart, but individual polymer strands, they form and they decay. So this is one reason that why the ectomyosin cortex is actually behaving more like a liquid on longer time scales, such as minutes. So one can consider it as a very viscous fluid if one probes the material slowly, so on minutes or tens of minutes. However, the basement membrane, that's a different story. That's behaving more like a solid-like material because it turns over very slowly, and the turnover is actually quite variable. So in embryos, it can be only a few hours. So in the Drosophila embryo was measured to be around 3 hours. In the wing disc, we found it to be on the order of tens of hours. And in fact, from human tissue, it is known the older you get, the slower it becomes. And I think if you are a little bit older adult, then it can be even years or tens of years that basement membranes are turning over. So this is exciting for physicists because we have this layer ectomyosin, which is viscoelastic and in fact liquidy for on timescales of many important biological processes, particular morphogenetic processes. On the other hand, we have the basement membrane, which does behave much more like a solid-like material and therefore also can bear elastic stresses. So that means if we deform the tissue, then elastic stresses in the basement membrane are generated. And if you release the external force, then the basement membrane would tend to bring the tissue back into its old shape. And we looked at this material properties of the basement membrane in the wing disc. In particular, we were interested whether there are mechanical tensions on the basal side, similar to like pre-existing mechanical tensions that we know from the active cortex. On the active cortex, we have active molecular motor proteins associated to its myosin motors, and those are the ones using ATP that generate contractile tension. In the basal membrane, there are no motors associated, but nevertheless, we found that also this extracellular matrix layer is actually under mechanical tension in the wing disc. So we looked at this by indentation, just regular indentation with pyramidal AFM cantilevers. And we initially had the idea to actually just do Hertz model indentation. Then we actually realized that those force indentation curves that we are getting there are not really looking like the typical force indentation curves that you would expect. So those should actually increase parabolically. So with the indentation, that's to the power of 2 for a pyramidal indenter. However, we saw that our force increases were rather looking linearly. So the Hertz model did not really match our observations. And that was hinting, or like looking at the literature, you can see that these linear increases is something that you would get if you are indenting in a pre-stressed sheet. So imagine, for instance, the sheet of a trampoline. If you would poke into that orthogonally, then you would have a force, or would need a force that would go linearly with the indentation depth that you achieve there. And that was then the basis for determining the tension in those basal membranes or basal sheets of wing disc epithelia. And we found indeed actually that the tension values are quite similar to what we knew from ectomycin cortex tensions in interface cells. And our study also showed that the tension that is generated there, so this is an elastic tension on the one hand. So it is present because the basal membrane is subject to an elastic stretch, but this elastic stretch is actually generated through the ectomycin cytoskeleton in other parts of the cell, so presumably in particular the apical phase. So the tension that is there at this apical part is giving rise presumably to a hydrostatic pressure inside the epithelial cell, and that then actually stretches the basal membrane so that we have even an epithelial homeostasis, so there has been no deformation of the tissue from the outside. Still, the basal membrane is subject to a mechanical stretch. We are also interested in looking into this in other tissue types, and this is work in progress right now.
That's amazing. Because also, like you said, if it changes with age, for example, I would also assume that it changes, for example, with inflammation or other disease conditions. So, for example, cancer, like you said, and kind of tissue stiffnesses. And you also picked up on one component of the cell, which is the actin cortex, which I personally absolutely love. And we obviously know that the biomechanics is super important for the actin cortex, and it plays a role in a variety of biological processes, such as, for example, cell division, migration, etc. And you had a recent paper where you actually spoke about the actin cortex, especially also with respect to the EMT, or epithelial mesenchymal transition. And you really characterized the actin cortex and how it changes during the cell cycle. So could you maybe share with us, what role does the actin cortex play in general? How can it play a role in the EMT? And how does it play a role in the cell cycle?
Yeah, so the actin cortex is something like on the scale of the cell, like the muscle of the cell. So it is a structure that can generate active forces. So the muscle in the body also contains a lot of ectomycin. So it's essentially the same structure in the muscle, but on a much bigger scale. In the cell, we only have a very small thin layer of this underneath the plasma membrane, so in a non-muscle cell, right? So in a somatic cell, like, for instance, cervix epithelial HeLa cells that we frequently use in our studies, or actually in this study that you're mentioning, breast epithelial cells. So if they are not adherent, then we have the actin cytoskeleton concentrated underneath the plasma membrane in the form of the actin cortex. If the cells are adherent, then the structure or the structures of actin are more diverse. We have stress fibers developing, and actin cortex, in addition, we have focal adhesions and so forth. So actually we decided to make things a little bit more uniform in terms of the actin cytoskeleton. We decided to measure non-adherent cells, and that also allowed us to determine surface tension in the study by deforming the cell away from this spherical shape, or actually from shape of minimal surface area determined by surface tension. So in this way, we could measure surface tension and as well the rigidity of this actin cortex shell at the periphery. And there we saw, not only in this study that you're referring to, but also in earlier work, that during the cell cycle evolving towards mitosis, we have an increase of the surface tension due to higher actomyosin contractility in the actin cortex. And that presumably gives a major contribution to mitotic rounding. So there is, of course, a second contribution, namely that the cell is de-adhering when it enters mitosis. So this would also contribute to rounding. Even for low surface tensions, the cell would get round if they were not adherent at all. So as long as it's not zero, surface tension would drive the cell into the shape of minimal surface area, which is a sphere. But if we still have a little bit of adhesion, then it helps if surface tension actually increases and then starts to dominate the morphogenetic processes of the cell and drives it into a spherical shape. So that's our reasoning. And during EMT, we see that changes in the actin cortex occur. So interestingly, opposite in interphase and mitosis. So in interphase, we found a softening and a decrease in surface tension, while in mitosis, the other way around. For several cell lines there, we have an increased actomyosin contractility, increased surface tension, and also increased actin cortex stiffness. And we could also see that this is likely because activity of certain molecules changes. So regulators of the actin cortex, Rho-GTPases, have different activity levels before and after EMT. So before EMT, we have, at least in the cell lines that we looked at, high Rho-A activity, so Rho-A rather than REC1. So we looked at Rho-A versus REC1 activity in particular. So before EMT, Rho-A activity was much more prominent. After EMT, REC1 activity was more prominent. Rho-A activity was downregulated. And that was contributing to these mechanical changes of the cortex. So in the interface, cells, in particular, REC1 activity increase leads to more ALP2-free generated actin cortex. And interestingly, this ALP2-free activity rise leads to softening. So although it polymerizes actin and can actually also enhance the abundance of actin at the cortex, this actually rather leads to a softening, which I find quite interesting and unintuitive. Was not published by us first, this finding, but by the lab of Claire Waterman, as far as I remember. So we could show this is also true for breast epithelial cells in our AFM assay. And we found that then in mitosis, actually there is the opposite trend. So possibly again, through REC1 activity increase, we have now a stiffening and more contractile cortex. And it is not quite clear why, because ALP2-3 also in mitosis rather softens the cortex, but in mitosis, REC1 has apparently other effects, possibly upregulation or activation of Rho-C. And through Rho-C, which is actually similar signaling pathways as Rho-A, the mitotic cortex under the rule of REC1 is becoming stiffer after EMT.
Because the thing is also, you looked at various different tissues, which I think is really interesting, because from a biology point of view, it's obviously every cell line, etc., or tissue has specifics that we need to consider. And you looked at different cell lines / different human tissues, which included breast, lung, prostate, epithelial cells. And so I thought it was really interesting to compare these. And maybe you can touch on why it is so important to compare different tissues and what you found in them.
Yeah, I mean, we first observed this effect that I just described, this changes of the mechanics of the cortex in breast epithelial cells. So there's also a couple of cell lines which is very well studied in the lab. So MCF7, MCF10A. But of course, we were interested in knowing whether this is a phenomenon that is conserved across several tissues in the body. And in particular, we were interested in whether it would maybe play a role in cancer. So EMT, epithelial mesenchymal transition, is believed to be a cellular transformation that kickstarts metastases, or at least can support the beginning of metastases, making cells less adherent to their primary tumor cluster, and then through this lack of adhesion, becoming more migratory. And our study suggests that it also has an effect on mitotic rounding. So through having a higher surface tension in mitosis, cells have the ability to become, even in a very crowded environment, such as a tumor, can still have round mitotic cells, which can then faithfully divide without chromosome aberrations and thereby still be viable, which is, of course, bad for the cancer patient, but good for the survival of the cancer cell. Since we wanted to know whether it has not only an effect potentially for breast cancer, but also for other kinds of cancer, we decided to look also into other cell lines from diverse tissue types, so prostate and lung in particular. And there, yes, we had the exact same phenotype, but I acknowledge that one should probably look at more cell lines from these other tissue types to confirm that it's not just a one cell line phenomenon.
But like you said at the start, I think there's so many different things to consider. If you even think of a single cell, there's so much complexity in it. And then to think about cells in the kind of tissue context and then compare different tissue cell lines, etc., it just adds to the complexity and what you could study, or just endless opportunities, which is good, but also obviously difficult. You can't study
a lot of
important way. Yes, absolutely. So kind of moving on from cell lines and different tissue types, another thing I wanted to talk with you about is actually different dynamics and processes. And you very briefly touched on it in the introduction, because you spoke about the fact that there's obviously fast and slow processes in the cell, and that the actin cytoskeleton shows obviously very different dynamics. So maybe you can talk with us about how your team actually developed a new technique to measure these actin cytoskeleton Poisson ratios and to understand how the actin behaves in these fast or slow processes.
So you're speaking about a study of ours, which was in collaboration with the lab of Sebastian Alland. Sebastian Alland, he is a mathematician doing numerical simulations based on the finite element method. And yeah, the collaboration with him gave the unique opportunity to look in very great detail how the actin cortex shell is deforming when put between two parallel surfaces, which are moving towards one another. So in this way, we called it cell squeezing assay. So essentially, we have the cell already confined from the start, so typically with a retracted AFM cantilever. That is then what we considered as our reference shape from an elasticity point of view, or also viscoelasticity point of view. And then we were bringing the cantilever a certain amount down, a small amount down, to deform this actin cortex shell away from this reference shape. And then not assuming from the start that we have an incompressible material, we wanted to actually measure two elastic moduli or viscoelastic moduli that characterize this mechanical system, the actin cortex. So, it's a bit challenging from a conceptual point of view to explain it. So essentially, if you start from different confinement heights in the beginning, so not just the retracted AFM cantilever state, which would be around 15 micrometer height, right? But also starting from already more confined shapes in the beginning with a lower cantilever height, then doing a small, we called it compression step, so uniaxial compression step, would deform the actin cortex shell in different ways. So if the cantilever is high, we start from an almost spherical shape, and then we have a combination of shear and bulk deformation of the actin cortex shell. And if the cantilever is low from the start, so we have already a very oblate shape, say, of the cell. So there's a small height, almost like a pancake. And then it moves more and more towards just a dilation of the cortex with very little material shear contribution. So depending on how we choose the initial height of the cantilever, we could essentially probe different modes of deformation in the actin cortical shell. And measuring then the force response of these uniaxial compression steps, starting from different confinement heights, we could essentially calculate the Poisson ratio of the cortex. So if one has the area bulk modulus and the area shear modulus of the actin cortex shell, then it allows you to infer the Poisson ratio. And then, as I said, one can also or should also consider the time scale dependence of cortical mechanics. So we did this in a time scale dependent manner. So we performed actually oscillations of the cantilever around a certain confinement height of the cell, either with a higher cantilever where the shape of the cell is closer to spherical, or around a more confined height, so lower height, where the cell is more pancake-like. And then we calculated for these different experimental settings what kind of material properties corresponds to the measured response, so Poisson ratio and an area bulk modulus, or vice versa, Young's modulus of the shell. And we found that we actually, for larger timescales, so slow probing of the actin cortex, slow deformation of the actin cortex, the Poisson ratio is indeed close to 0.5, as everybody usually simply assumes in the publications. This is a priori not at all clear from my point of view. But for faster timescales, so 1 hertz and faster, the Poisson ratio is lower and rather goes into the range of 0.3, which would be very typical for a polymeric material. And but this may be also interesting. So the experimental data say that maybe the Poisson ratio is even a little bit larger than 0.5 at slow timescales, which might be a little bit confusing initially, but if one keeps in mind that this is not necessarily an isotropic material, so we can have polymer orientations in the actin cortex, for instance, a bit more in plane, so more tangential, maybe rather than completely isotropic. And then we can have actually values of the Poisson ratio greater than 0.5. Actually, formally, for 2-dimensional shell, one can have values up to 1. And we measured something like 0.6. But to be fair, given the error bars, I guess we cannot be certain whether it's really greater than 0.5 or whether it's simply the error bars that add on top.
Again, so many considerations. So interesting to hear you talk about the research and all the different layers that need to be considered and kind of pulled apart. And again, so many kind of disciplines as well, like the modeling, the physics, the biology, everything needs to be so well understood to really get an insight. So it's really interesting to listen to you. And I guess there's so much more research to be done. And unfortunately, we don't have time to talk about all of it, which we would love to do. But, time is always limited. So I just wanted to ask you like a last question, which is, what are you most excited about in your research?
Oh, what am I most excited about in my research? I have to say, I love doing research because I love doing or solving riddles. That's what I realized. So I'm a very curious person, and I love it to look into riddles, I guess. And my riddles are biophysical riddles. That's why I became a scientist. And about this particular subject that I decided for, I guess, is to understand forces and compliance of forces, and to combine it with the aspect of life. So, and with things that you cannot all touch them, admittedly, but at least you can touch tissues, you can draw analogies to your own body, to dynamics that you see in your own body. I guess this is a particularly exciting aspect of it.
Absolutely. And as we kind of really heard a lot about today, there's always more riddles to resolve. And I think that's also the exciting thing, that when you solve one problem or get insight into one result, there's always 5 more follow-up questions. And that's great. So that you will never run out of work, which is amazing. So,
yeah, that might be a threat for some people. They never run out of work. But I guess scientists are, in fact, quite happy about it.
Yeah, you have to keep it fresh, right? So it's always good. Science is never done. There's always more to do, which is great. And so to our listeners, this is all that we unfortunately have time for today. But we have heard so much from Professor Fischer-Friedrich about her work, and how her work really spans various aspects of fundamental biomechanics, from the actin cortex to the basement membrane. We have spoken about tissue cell lines, we have spoken about cancer, we have also spoken about Drosophila wing discs. So we have really covered a whole lot of different aspects. So firstly, thank you so much for your time, Professor Dr. Fischer-Friedrich. It was great to have you here.
Thank you very much for having me. It was a pleasure.
If you haven't done so, please be sure to listen to our episode 5, where we have talked to multi-award-winning Dr. Craig Prater about nanoscale chemical analysis using AFM IR. So please be sure to stay tuned for more, and we will see you next time at Conversations on AFM. Thank you. Bye.
This podcast was sponsored by Bruker Nanosurface and Metrology Division. Bruker is a manufacturer of scientific instruments for life sciences and materials research, as well as for industrial and applied analysis. If you'd like to learn more, please visit www.bruker.com. This podcast was produced by Keep It Science.